if there are questions that would come up. Everyone involved in this challenge. Okay, so while they're coming up, does anyone have a question? Yes.
Yeah, hi, really nice presentations. And I just had a couple of questions actually for each of the last two presentations. So in the last one, I'm wondering, how do you deal with the fact you've tested so many kernels in the cross-validation step or in your evaluation? Is that something you considered, or is that something that needs to be somehow factored in, and how would you do that?
Well, the question is how do we finally choose the right combination?
So certainly each kernel combination of parameters. I mean, the kernel itself is essentially a parameter, right? And so each combination of parameters gets a score. But I'm wondering if you wrap that whole thing inside of some kind of cross-validation framework, or you somehow correct for the fact you've tested. You've given yourself so many chances to succeed just by different combinations of kernels. Right. Each extra kernel is another hypothesis test you're doing, basically.
Yeah, I'm not sure. No. I mean, it was, well, given one. It's independent cross-validation testing, so...
For each one, right? And so I'm wondering, can you wrap the whole thing inside of a cross-validation framework?
We didn't try that. We didn't...
Because it may be that if you did that, maybe you'd... I don't know if you draw different conclusions, but you could maybe push up the performance.
Maybe it's a good idea to average all the... Well, to aggregate in some ways the different results of the different combinations, but we didn't try, but it might be a good idea.
However, the last result were, maybe that wasn't clear, but the final result was done in a completely independent test set.
Oh, absolutely. I mean, absolutely.
Right. But in the training,
at the end, but there was the one that did number 2, and then there was the one that did number, was it 19 or whatever? Yeah. I mean, maybe that would be the one. I don't know. That could improve.
Yeah, and also on the leaderboard, we were completely out of the first. So just to say that on this challenge, at least, it seems to depend on the nature of the test set.
Yeah, thanks. And then just one last question for the Southwestern group. Really nice, clear presentation. I'm wondering about your decision early on to throw out SNPs that were synonymous. Because my understanding of a SNP is that it's really just a marker for some other polymorphism that's the real variant that causes the disease or the condition. I'm wondering what would happen if you try just including all, not having that filtering step.
So the decision to throw out those synonymous mutations was very early. Actually, I didn't try whether keeping them will make a huge difference or make the model better or not.
Yeah.
So probably I can just give a little bit thought on this, because really for us the main problem is that we have too many SNPs. We know that probably we will lose some information by throwing out some of the SNPs, but I think probably we will reduce the noise level much more than reduce the signal level. So for us, really, because there's so many things to try. So for us, I think the first thing is to try to reduce the noise as much as possible. So that is just the intuition behind that decision.
I mean, I'm guessing, not knowing what the exact numbers are, I'm guessing that just throwing out SNPs outside of genes or far from genes took care of a lot of it. I wonder.
Yeah, the original one is 1.6 million, and then after that step, we throw about half of the SNPs. So 0.6 million.
Yeah. So the remaining SNPs are more likely to be functional.
Yeah.
I mean, I would have thought that would have thrown out 98% of the SNPs if there's 2% of coding region.
Sorry?
Well, I suppose I would have thought, since genes make up 2% of the genome, that you would get rid of 98% of SNPs by excluding SNPs not in genes. Thank you.
Actually, you repeat that so that everyone's
Right, so Ivan, what you said is that it was a random genotyping?
The SNP arrays are not made for random SNPs.
Right.
I don't know if that's quite right. Well, but you shouldn't have
Try to come to the microphone.
sorry. We just have to look into it. They're not random, but of course, if you exclude all SNPs that are not actually coding, I think you'll be getting rid of far more than we're gotten rid of. So maybe you mean within sort of gene boundaries. Coding is different. So, okay.
I have one question. It's interesting to see that the Southwestern group did so much biological screening first before choosing the features, and then eventually you got to the machine learning, whereas the CASI group seemed to have done mostly data-driven without much biological thinking beforehand, and that you got very good results. I think tells that there are room for the two approaches. But for the CASI team, why didn't you look into more biological thinking into your algorithms?
Well, first of all, because this method has shown good performances on related problems, so it was motivation to try it. And then, when you say that it was not biological driven, it's true that we didn't select it a priori some SNP or some genes, but maybe we think that, for instance, for building unkernel drugs, you have to build some descriptors, and these descriptors are really important. So, for instance, we didn't try the dragon descriptors on chemical, and maybe that would be a good idea. So it's not biological driven because you select a priori something, but you still have to choose your descriptors. So, for instance, SNP for cell lines, and maybe we included RNA-seq information, but and we didn't see that, and that it was not we saw it was a good idea to include it, but maybe it would be also good idea to add some epigenetic information or so. So you combine all this information, but these information are important, and these are biologically driven.
I have a question about TUBA-UT Southwestern Group. Hi, that was a great job. The question I have is, you have the geographical component was giving you small prediction accuracy, and also the principal components based on SNP decomposition was giving you small values. But when you decompose SNP data like that, usually you get ethnicities. How much was the principal components correlated to ethnicities from the cell line data? Because both would be, in a way, correlated to ethnicities.
So I'm sorry. So how much? What's the last sentence? So how much the principal component contribute? Can you repeat the last one?
So when you take a big matrix of SNPs and you decompose it, usually what you have is like principal component 1 is African Americans, principal component 2 is Caucasian, principal component 3 is, Native American, so forth. So, did you see that kind of structure?
Yeah.
Oh yeah, maybe we can.
We do have some data, or you can just describe it.
Can you put this slide?
So probably we can just describe it.
Yeah. So after our dimension reduction and MDS plot methods, we ended up with like 100 principal components. And if we plot a plane of PC1 versus PC2, we do see those cell populations clustered into different groups. However, the way we screen those SNPs, during that process, we make sure that the remaining SNPs carry some kind of functional data there. So the cluster, so the principal components not only carry the geographic area information there, they also kind of carry functional information there, which helps us make the prediction.
Yeah, I'd just like to add that so the first few genotype PCs were pre-computed by us and removed from the effects on the EC10 to begin with. So in other words, the information that was useful here. It may cluster geographically, but would be, maybe down at the level of 5th or 6th PCs. The first few dealing with continental ancestry were kind of pre-removed. And so it'd only be sort of nonlinear or lower PC effects that I think they were able to extract usefully. And also sex was also regressed away, which I think explains why that wasn't helpful either.
So one last question?
Thank you.
I just have a quick question, largely about the data for both groups. Both groups noticed that there was some batching in the data. Where was it coming from mainly? I think I got a little confused by the data set. Is it mainly the chemicals, the SNPs, or something else? And also, is it possible, do you think, to address some of the batching at the point of data set construction rather than in the algorithm?
Let me attempt first, and Yvonne Rusin can add if I say something wrong here. So the nature of this volume of cytotoxicity profiling required that the cell lines be grown up in batches and then done in a series of screening steps and plates at the NCATs. It's a fancy robot doing all this stuff, but still the throughput is always going to be limited. So there were 5 or 6 major cell line growth batches. Everything was randomized beforehand in terms of the sex or gender and the continental ancestry. So a lot of things were balanced beforehand, but at the end of the day, these were done in about 6 batches. And so those level of batch effects, there were, in terms of linear effects were pre-removed, we removed the batch effects through essentially using COMBAT, which is designed for microarray analysis, and does a fair job of making, for example, PCs overlap. However, if you look at other kind of orders of the structure, you can see them still very clearly. So is there anything to add? Okay, so apparently that's correct.
So the remaining batching was in the SNPs still?
I don't know if, when they use the term batch, if they were referring to batches of cell lines.
Yeah, exactly.
Yes,
Yeah,
okay.
So what was shown was clearly ancestry effect and also some chemical structure. So those are things that are come with the population or the actual chemical that you will screen. So the actual technical variability and other things that we call batch effect were removed. And it was pretty easy to remove because it was very systematic. And this is fluorescent detection, so it's just like microarray. The reagent concentration on the detection stage will vary, experiment to experiment. So it wasn't major. We really were not concerned about that. The data was highly reproducible, and we're happy to talk about that offline.
Okay, I think we should go into the awards now. Before we do that, and before everyone leaves, just want to make sure that there are a couple of other people who were not formally introduced. I refer to the Yang Lab. So this is the Yang, Dr. Yang C. From the Yang Lab, and also Yvonne Rusin, who was very conceptualized the experiments with Fred and did the experiments. So he was a major contributor to this as well. So I think we've got a complete set of introductions. And Gustavo, do you have these are the $100,000 checks? Oh, no. Okay.
Checks are in the mail, right?
Better yet, we have certificates.
Right. So let's start by.
Should you come to the microphone and actually, if you
Yes.
announce it, I'll.
Very good. So prepare the camera. Team Yang Lab and Team QBRC are the 2 best performers of 1 and 2, and we start with that. Thank you.
Okay.
Is it? Why don't you guys go together there, take a picture. What do we need? For now, yes, and then we will go with the other one.
Come to the front. I can go down the steps.
Thank you. Excellent. Thank you very much. And now, second best performer in sub-challenge 1, Team Cassie, and Elsa is going to receive. Thank you.
Yeah.